Now that we know the parts of the cell and what each of those do, it's time for us to see how these parts come together to make the cell function. This is known as cell physiology. There are three pieces of cell physiology we want to examine: cell growth, cell reproduction, and protein synthesis. Cell growth and reproduction occurs in what is known as the cell cycle. The cell cycle is primarily interphase. In interphase, the cell is growing, it's performing its normal functions, it's just doing what it's supposed to do as a cell. At some point in time, if this cell is going to divide, it gets a signal that it's time for it to divide. And at that point, it makes copies of the DNA and all of the organelles so that when it divides, it will have enough material to go into two cells. Mitosis is the process of the division of the nucleus. Now this gets tricky because we have DNA, we have chromosomes that we have to make sure each new cell gets one copy of the original DNA material. And so mitosis is a little bit complicated. But this is the separation of DNA so each new cell gets a copy of the DNA. Cytokinesis is the very last step. We have to divide the cytoplasm and the organelles in the cytoplasm. At the end, we will have two new cells that are identical to each other and identical to the original. These cells will go into interphase and stay in interphase until it's time for them to divide. Most of the time, the cell is in interphase. There are three stages of interphase. The G1 phase is the growth phase. This is the cell growing and getting larger. It's just divided, so it has to take on some growth. It has to get a little bit bigger. Then there is the synthesis phase, this S phase. This is when the cell knows it's going to divide, and so it replicates the DNA. And then the third phase is this final last growth spurt. The cell gets a little extra large. It's going to have some extra cytoplasm to make sure it has enough cytoplasm to go into the new cells after it divides. DNA, you'll remember, is deoxyribonucleic acid. It's made of subunits called nucleotides, which are composed of a phosphate, deoxyribose, which is the sugar, and a nitrogen base. And there are four possible bases: adenine, guanine, thymine, and cytosine. So a single strand of DNA will have the phosphate deoxyribose backbone, and the bases will stick out to the side. Now DNA is structured as a double helix. It's double-stranded. We have two strands of DNA and they hydrogen bond to each other. They hydrogen bond at the nitrogen bases so that the two strands are held together. Not any two nitrogen bases can connect up. We have what's known as complementary base pairing. Adenine always pairs with thymine and guanine always pairs with cytosine. So you can see adenine thymine, guanine cytosine, adenine thymine, guanine cytosine. That's always how they pair up. This is very important in the way we copy DNA. During DNA replication, DNA makes a copy of itself. First of all, we have to uncoil and unzip that double helix, and there's an enzyme called helicase that does that. Then we have complementary base pairing occurs. This makes sure that we make an exact copy of the old DNA molecule and we end up with two identical DNA molecules. There are other enzymes called polymerases that go out and find the right nucleotides and bring them up to the exposed part of the DNA. These nucleotides are held in place until they can be joined to each other, and we will get two strands of DNA, so that each new cell has DNA that is half old, half new. Here we see DNA replication going on. DNA polymerase is the enzyme that's bringing the nucleotides up and having them hydrogen bond and holding them in place until those phosphates and sugars can bind to each other. Because of complementary base pairing, we'll always get an exact duplicate. Adenine always binds to thymine and guanine always binds to cytosine. We say we use the old strand as a template. That is, the old strand is the pattern, and we make the other half against it, again, using that complementary base pairing. So this is part of the old strand, and over here is part of the old strand. And we're bringing up the new nucleotides and attaching them in the right order so that in the end we're going to have two strands, half old, half new, but they'll be identical to each other. There are special enzymes that go through and proofread just in case there was a mistake made so that corrections are made before the cell divides. Now that we have the DNA replicated, we can start the mitosis or M phase. This is the nuclear division phase. Those replicated chromosomes have to be divided. We have to make sure that each new cell gets one copy of each chromosome. There are 4 stages of mitosis: prophase, metaphase, anaphase, and telophase. In prophase, we see the chromatin condensing. Normally the nucleus is kind of invisible. It's got a membrane around it, but you don't notice too much going on in it. But during prophase, the chromosomes start to condense and you start seeing strings of chromosomes rather than just kind of an amorphous material. The nuclear structures disappear. The nuclear membrane disappears, and we start to see the chromosomes just sort of dumped out into the cytoplasm. The second stage of mitosis is metaphase. During metaphase, the chromosomes line up at the equator, and we see the centrioles. They're anchoring themselves to the cell membrane, and they're sending out spindle fibers to each chromosome. During anaphase, those spindle fibers are going to shorten. Now, these identical chromosomes had been held together by something called a centromere, and at anaphase, the centromere divides so that the chromosomes are no longer held together, and as the spindle fibers shorten, they're pulled to opposite poles of the cell. The final phase of mitosis is telophase. During telophase, the spindle fibers begin to disappear, and we have all of the nuclear material down in each end of the cell. We begin to see the chromosomes disappear and the nuclear membrane reform. Cytokinesis begins sort of simultaneously with telophase. We start to see the cell membrane pinch in, and it's going to get closer and closer, and when it touches, we're going to divide and we're going to have 2 new cells. The nucleus will completely reform, the spindle fibers will disappear, and we will have 2 new cells exactly like the old cell. Genes are simply specific segments of DNA that contain the instructions to make a particular protein. So the DNA has to carry some sort of genetic code. We have to have some way of denoting which amino acid goes where on a protein strand. The genetic code is composed of 3 nucleotides. Now, when these 3 nucleotides are part of DNA, they're referred to as a triplet. We usually refer to the genetic code by using the letters of the nucleotides, A, T, C, and G in the case of DNA. Now, the DNA contains the master blueprint for everything the cell can do, and that is kept in the nucleus. But the protein factory is out in the cytoplasm. You'll remember that the ribosomes are involved in making proteins. So we need to get those instructions to the production site. Plus, we don't need to take the whole blueprint. Nobody ever takes the whole blueprint to the construction site. They always keep the master copy back at the office. So we only need to copy that piece of the blueprint we want at the time, that one gene. We're going to use RNA to do this. Now, there are 3 kinds of RNA: messenger RNA, transfer RNA, and ribosomal RNA. Ribosomal RNA makes up part of the ribosome. When we start converting DNA to RNA, the process is called transcription. We're making a copy. The section of DNA that has the protein, the gene on it that we want, unwinds a little bit, and we get that gene copied as RNA. Now, this is going to be messenger RNA because it's going to take the message out to the ribosome. You'll remember that RNA is single-stranded, and instead of thymine, it has uracil. Also, its 5-carbon sugar is ribose and not deoxyribose. When we make messenger RNA, we do it just like we did DNA replication, but we use RNA nucleotides, and of course there are some different enzymes involved. Here the complementary base pairing is adenine with uracil. Uracil replaces thymine. Cytosine still pairs with guanine. The triplets of DNA, when they are transferred to RNA, start being called codons. Since DNA can't leave the nucleus, RNA can. This is how we're going to get the information to the ribosome. So here we see a piece of the DNA unwound. We're going to have nucleotides, and this time we're going to have the enzymes bring up the RNA nucleotides and precise base pair them. Adenine will be pairing with uracil now instead of with thymine because there is no thymine in RNA. And we'll get this strand of RNA that is going to carry the genetic code for that one protein. The triplets here will be converted to codons here. Now, messenger RNA is made from DNA, and each 3 nucleotides is a codon. This shows the genetic code. Each codon codes for an amino acid. It turns out we have more code words than we need. So we say this is a redundant code. Here you can see all 4 of these codons will code for this one particular amino acid, which is leucine. And you can see that there are plenty of duplications throughout the genetic code. This AUG stands for methionine. It also tells the ribosome where to attach and where to start. And then we have 3 codons that do not code for amino acids, but rather they tell the RNA to stop adding amino acids at this point that we're at an end for the protein. Amino acids are the monomers of a protein, so this is what we need to build our protein. Protein synthesis consists of 2 basic steps. Transcription, which we've just looked at. This is making the messenger RNA in the nucleus using the DNA as a template. The messenger RNA can then leave the nucleus and go to the cytoplasm. Once it's in the cytoplasm, we can begin the second step of protein synthesis, which is translation. In translation, the rRNA, which is part of the ribosome, attaches to the mRNA, and we have the ribosome forming around the messenger RNA. The ribosome then reads each codon. It looks at each 3 base pairs and determines which amino acid that's for. The transfer RNA is the RNA that has the amino acids attached to it. Now they will come up, and again, using complementary base pairing, the transfer RNA carrying the correct amino acid will bind to the messenger RNA in the ribosome. The tRNA has what we call an anticodon, that is the opposite of the codon, so that the correct amino acid is added to the growing protein chain. So transcription goes on in the nucleus. We unwind a little bit of the DNA to make the messenger RNA against it. The messenger RNA can leave the nucleus and come out to the cytoplasm where it will attach to the ribosome. As the ribosome is reading each codon, transfer RNA is bringing the right amino acid and holding it in place until we can bind the amino acids together. Whenever we hit one of those stop codons, the ribosome drops off and we're done making that polypeptide chain. We've talked a lot about a general cell, but we need to realize that cells have a lot of diversity and cells specialize in what they do. When they do specialize, they may lose some of their general characteristics. For example, we have cells that connect body parts. These are fibroblasts. They're very interested in making thread-like proteins that will hold other cells together. Also, erythrocytes help connect cells to each other. They carry oxygen to other cells. And erythrocytes have lost their nucleus. This makes them smaller and more flexible and allows them to go more places. Certain cells are specialized to cover organs or line tubes. They have very tight junctions between them that does not easily let stuff get through. Other cells move organs or parts of organs. They're capable of contracting or changing shape. This would be skeletal and smooth muscle. Some cells store nutrients, like the fat cell. It stores fat globules. These are called adipocytes or fat cells. Other cells may help us fight disease. The macrophage is specialized to eat other stuff, so anything foreign it will eat and take care of. Certain cells control body functions, such as the neuron. The neuron is capable of conducting electrical impulses. And then we have cells for reproduction, like the egg and the sperm. And here you see a sperm. The sperm is one of the few motile cells in the body because it has to swim to the egg.